Now it's my pleasure to invite our second speaker, Dr. Anuranjan Anand from JNCASR, Bangalore. He'll be speaking again on a very important topic, genes for sensory epilepsy, which are triggered by tactile and temperature cues. Dr. Anand is a faculty in Molecular Biology and Genetics Unit and an adjunct member of the Neurosciences Unit at JNCASR, Bangalore. The Department of Biotechnology of the Government of India has awarded him the National Biosciences Award for Career Development in 2008. Indian Academy of Sciences has elected him as Fellow in 2011 of the National Academy of Sciences, India, and in 2013. And he has also been elected as a Fellow of the Indian National Science Academy in 2016. So, sir, please give the lecture.
Thank you so much. A lot of introduction gets covered. She speaks almost 3 times faster than I can speak, and with lots of names of genes which look like, email passwords and Wi-Fi codes, DJ1 and Chacha D2, etc., etc. It makes me very nervous. So thanks, Thelma. And the questions that you have, I think if they have not been answered by Thelma, will be answered by Ganesh, who talks after that. So I'm very appropriately positioned between. So thanks to Professor Keshav Singh and Thangaraj for inviting, and very imaginative, appropriate title, Human Diversity and Health Disparities. The more I thought about it, by the time it sunk in that I have to speak here, I think this began about a year ago, I think my lungs begin to explode because it packs a lot into it. And the only via media is mentioned about Analaba and Thangaraj, and take a shortcut to what you're going to talk there. But I think it requires, given the genomic divide that can be set in, it requires co-culturing of many disciplines, basic biology being one of them, but interfacing with a lot of medical specialities, human biology, and appropriate policy making if benefits of all that we are talking about now. And we'll talk about how to percolate down to things. It's a big topic, actually. So thanks for organizing. I haven't seen a title like this, so encompassing earlier. I'll be talking about something which began as a fun activity in my lab. Epilepsies, as most of you know, are common brain disorders. Lifetime worldwide prevalence is about up to 3%. It's not a single disorder, but a heterogeneous collection of disorders. There are about 40 types of epilepsies. The most common characteristic of each one of them is seizures, which are transient signs of abnormal excessive synchronous activity of the neurons. It affects people in childhood and in older ages, though etiologies are very different. In very difficult situation, surgeries can be attempted, etc. Now, whether you look at it at a single cell level, which is a very limited view, or you look at a neuronal network level, which is a more appropriate view, there is substantial genetic basis to etiologies of epilepsies. Genes are involved. Up to about 10 years ago, we didn't know etiologies of about 2/3 of the epilepsies, but that's begun to change after some hard work done in 90s using classical genetic approaches, following by a rather unproductive and disappointing phases of GWASs, but more so now with whole exome and whole genome sequences, which are large collaboratives coming into place in the world, illuminating the genetic causes of many of the epilepsies, like localized ones, which were considered largely acquired or environmental, are turning out to be having genetic cause there. I won't dwell on nuances of each of these. They have limitations. When you look at cell or network, the heritabilities are strong. In this case, it's very intuitive because genes get involved. But if you look at EEG patterns, it's close to about 80% heritability, indicating that genes are going to play major roles in causation of these. One class of very curious epilepsies, very intriguing epilepsies, are sensory reflex epilepsies, where epileptic seizures are triggered by defined and controllable factors. The most popular of those, most common of those, is the one which is triggered by visual cues, visual stimuli. And one of the most dramatic example of this was reporting of a large number of children with photosensitive epilepsy after the Pokemon serial was released in Japan. Apparently one of the characters, Pikachu, has blinking lights in his eyes, and a large number of children develop visual epilepsies. There's much work, a few loci known, perhaps no gene. Auditory epilepsies are again a common class. Some of the genes have been found in a very curious way. For example, in mice, when animal houses are being cleaned with high noise making vacuum cleaners, some of the mice develop seizures, led to very nice stories and discoveries of genes later on found in humans. To me, one of the most intriguing class of epilepsies has been which are triggered by touch with lukewarm water, 40 to 42 degrees. I didn't take this phenotype very seriously for a long time, but more I thought about it, more it blew me off. If a simple touch of hot water can trigger seizures, there must be some level of curious biology behind it. I must mention that the normal brains do not have pathological responses and are able to handle whether they are flashing lights or loud sounds or touch with hot water, where essentially each of these sensory modalities, here skin is the ear or the eyes, trigger seizures or one or the other nature. Now, another reason of interest in hot water epilepsy was, which something Thangaraj alluded to in one of his slides, where he mentioned about Handegodu and Madras motor neuron diseases, things which are very, very, perhaps not very specific. We give them a name and call them specific, but I think there's a high prevalence of that in certain pockets in India. There may be overlaps, phenotypes elsewhere too. So with respect to hot water epilepsy, while the very first case was from New Zealand in 1945, among 21 cases of reflex epilepsies, one case reported of hot water epilepsy. There have been reports from other parts of the world, as you can see here, but close to about 90% of the cases, about 2,000 cases reported so far, are reported from India. Studies beginning in 90s in NIMHANS and continuing to date, where hot water epilepsy patients are reported and are admitted pretty much every week, are from limited geographies in India too. Now, just to rub the phenotype a little bit more for everybody, here is a case found in Saudi Arabia, where after pouring hot water on the head, ritual bath, the child suddenly becomes floppy and develops generalized seizures. Again, case from Belgium, where loss of consciousness and uncontrolled movements of the upper limbs while taking a warm bath. This is a curious case where grandparents hailed from India, generalized seizures while the child, 11-year-old Saudi girl, is taking hot water bath, and a case of reflex epilepsy due to warm water bath in 6-year-old. So it's not something which is just imaginative. Indeed, there are cases. A number of neurologists are intrigued by these epilepsies. The epileptic attack is sudden and occurs without warning. And if you step away from the clinical angle, the research on perception of touch and temperature sensation, to which the key phenotype of hot water epilepsy seems to connect to, is interest among many biologists. They'll be keen to know what molecules are involved and model them in organisms and things like that. So this is the phenotype. You can go through it. Dazed look, sense of fear, as if something bad is going to happen, irrelevant speech, hallucinations on the interface of neurology and neuropsychiatry. Frequency of seizures depends on head-water bath, at least in the families that we examined, seizure onset, and there's an element of self-induction, OCD-type behavior, which precipitates hot water epilepsy in many of these patients. Most of the patients come from certain regions in Karnataka called Mandya. I never understood why, but this is what is told to me. Perhaps it's a little more common, but maybe it's geographical excess of NIMHANS that makes it possible, etc. So I wasn't sure how to go about it. It took a very safe route, examining large families. At least when the phenotype is consistent among many members, hot water-induced seizures. There are multiple affected members. I can take a swing and try to find out what molecules may be involved and develop stories from there further. There was also a sense that there may be founder effects, which didn't hold out to be true. For a very long time, people didn't believe that indeed there's a clinical entity called hot water epilepsy. And it was thought of as a variant of febrile conversions, where fever-induced seizures in children are found to be, genetics of which is well explored. So some of the work we have done touches upon some of these things and partly answers as to where we are heading with respect to the clinical phenotype and genetics of this. Okay, this is only one slide which tells you what we have done. We put in about, it serves two purposes. It tells me about what we do. It also tells the age of investigators, because no longer people use microsatellite markers. The people who have used these markers must have begun their career in 90s kind of thing, and then paused for a long time for exomes and genome sequencings to come in. So I belong to that era. So we put in a lot of markers, about 500 of them, not lots by today's standard, on a family. Examine each of the members. A family with these markers, you don't need better density than this. Each of these is about 10 centimorgan or 10 megabase intervals, and then find out whether phenotype is linked or not linked. So each marker tests the probability of linkage as opposed to non-linkage, and 2 or multi-point lot scores are calculated using a very simple pipeline. Pipelines begin much earlier, though we talk about them now. And there's a stat put in which lets you exclude or include regions with fair amount of confidence. So we essentially did this for about 5 families and found 5 regions at location chromosome 4, chromosome 8, chromosome 9, 2 regions on chromosome 10. 2 genes, one G protein coupled receptor and SYN1, I'm forgetting what the full form of this, have been reported by others with similar phenotypes but minor age differences compared to what we are looking at. So after doing gene mapping, what we essentially do is come down to a very small region of the human genome on a particular chromosome, wherein we have excluded more than 99% of the genome, 3300 megabases, and direct all our efforts on less than 30 MB of the region. So in each of those families, we whole exomed and then whole genomed, which is underway for 2 of the families, and at least 2 individuals, and examine not only the candidate genes but all the regions. That tells us if there's a segregating allele, and then all other alleles which are not segregating, if eventually there's to be a single mutation or variant which is to be segregating. Most of those families, as you can understand, are indicated of single major gene effects to begin with, so that's our hypothesis. Now, what I'll do now is to tell you about Ggrf, a gene we found in one of the families which mapped to chromosome 4, R3HCC1, another gene, and SLC1A1. These 2 we are not very confident for reasons, one because of technical reasons of sequencing, and other for the variant that we have found is relatively high prevalence in about 1 in 200 in controls. So I'm breaking my head to figure out whether we are on the right track or not. But we initiated genomes here, and perhaps we'll find out if there are other things in these families segregating or not. With respect to febrile conversions, I mentioned about, there are 9 locations at a time this slide was made. Now there may be 2 or 3 more. And these are hot water epilepsy loci that I indicated, and 2 others which I haven't added. Apparently, shown in maroon here are febrile conversion loci, and the green are hot water epilepsy. It turns out that none of the hot water epilepsy loci are febrile conversion loci, and vice versa. So far, there doesn't seem to be any overlap of genetic etiologies of what is the causative for febrile conversions, and hot water epilepsy stands as an independent clinical entity as to what it does to the patients. Now, this is a family. It has a number. We found very good genetic score, lot score of 3.50 for a marker, defined proximal and distal boundaries at this particular location. You can see haplotype segregating among affected members, since most of these scores peak at about 70% penetrance value. That tells you about what is the impact, biological impact of, genetic impact of that in the particular family that we have examined. Now, we found in Ggrf this particular change at the protein level. This is the change. At this stage, we take that gene and examine in 288 hot water epilepsy patients from the same population. So they have been sort of very carefully examined, recruited in for homogeneous phenotypes, and we found additional 5 rare alleles. It's not that they are absent, some of them are, but some of them are present at about 1 in 1,000 or lower frequencies. So we find additional alleles. So this shows the conservation of the amino acid residues and traces of sequences, telling us that this gene, we have 6 variants found where the gene is, what the protein is. The time we found this gene, it was a very fragmented gene in the database about 3 years ago. There was no cDNA available. So we begin to communicate with bioinformatics centers. After some time, the cDNA became available, and the centers reached a consensus that indeed there's a gene exists. Both these happened in the right sequence, giving us a confidence that indeed the gene. Earlier it was a number, C4ORF21. Now it has a name, Ggrf1. And these are all bioinformatically deciphered domains. Not that we know from protein studies. So there's a helix domain, there's a nucleic acid binding protein, and there's a domain which I don't remember what it is, DUF2439, but it's in yeast in those genes which are important for cell cycle regulation. The locations of mutations are shown here, and the gene is expressed in certain brain regions in the humans at transcript level, and at the protein level there's immunoreactivity with respect to anti-Ggrf antibody. And if we see its cellular localization, this is done in HEK293 T cells, but we use at least 3 cell lines to look at localization. Is important, otherwise it can be very subjective. Ggrf apparently localizes the nucleolus, the centrosomes, in the spindle poles, and to the mid-body. So all these, in addition to a generalized staining elsewhere. So all these are indicators of its role in cell cycle somewhere. And I think this gives another view of its localization in the same entities. And apparently some of the mutations do cause mitotic defects in heterolog. And many of these assays, I should warn, are subjective to the cell line we use. So doing them in multiple cells and eventually doing this in a better animal model is imperative in these cases. But so far, the hint is towards that something in the cell cycle may have gone wrong in hot water epilepsy. I don't know. Maybe it's a developmental molecule and it embeds hardware defects right in the development. And later on, when challenged due to hot water, the brain is not able to handle volume of signals which go to the brain, and you see the phenotypic manifestation. Is one of the crude working hypotheses that we have so far. Another family mapped to chromosome 8 location, we found the gene called R3HCC1. It is an RNA binding protein. It has 2 domains, R3H and RRM1. 3 mutations found in this particular gene. High homology in mouse, identity and similarities are shown here for the domains and for overall protein, and with Drosophila, hinting to us that we could use either Drosophila or mouse to try to model hot water epilepsy. I think we're keen to explore that in humans right now much more. I don't know what hot water epilepsy will mean to flies. It's expressed in the nucleus and in cytoplasm. It localizes to stress granules. These 2 markers, PABPC1 and G3BP1, are canonical markers for stress molecule. And there's a high co-localization under 2 temperature stress conditions and 1 chemical stress condition of these markers with R3HCC1, giving us one biological hypothesis as to the direction we can take to explore in cells further. To develop a somewhat better model, we are beginning to touch primary hippocampal neurons in a mammalian system, and R3HCC1 indeed is expressed in somatodendritic cells, locations, and this is one of the neuronal markers, and you can see to what extent they localize or do not co-localize, and in the processes of the neurons where R3HCC1 is expressed. in these primary neuronal structures. The third gene is in family 300 at chromosome 9. The gene has a name, EAT3, excitatory amino acid transporter 3. Some details are given here. This is a schematic. That's the conservation and the trace showing you, and the co-segregation. Is not found in databases, mostly absent in 500 control chromosomes from normal individuals. EAT3, there are 5 transporters. One of them is expressed in eye and other at either pre or postsynaptic neurons. So this is a tripartite synapse, pre and post and glial cell. So it's one of the transporter of one of the common neurotransmitters, postsynaptic neurons. It's located largely glutamate synapses, to some extent the GABA synapses too, and these are responsible for reducing extracellular concentration of glutamate from as high as 1 millimolar to several orders of magnitude decrement. And the mutations that we have are shown here in the previous slide, 1, 2, 3. And if one does glutamate uptake assays using a protocol in C6 glioma cells compared to the wild type, there's a decrement of glutamate uptake for each of these mutations at 2 temperatures. Yes, a little worse than the other, suggesting that these are loss of function mutations. While one can't do standard electrophysiology, there's an ion which get co-conducted when the transport of glutamate takes place, and there's a decrement in electrophysiological parameters too for each of these mutations. So the take-home message here is that 3 mutations we have found are partial loss of function mutations by electrophysiology and glutamate uptake in a gene which could do much more, perhaps if it's deleted or has substantial damage to it, which seems to be true. It lights up in Alzheimer's and schizophrenia, etc., etc. We catch it through a hot water epilepsy phenotype window. So this is a spectrum of excitation by glutamate. Its toxic effects are kept under control because of very efficient mopping up mechanism because of EAT molecules which I indicated. Otherwise, even at 1000 to the concentration that occurs in the brain, if you do it in cultures, there's a huge amount of cell death. So obviously it needs to be packaged and taken away to avoid excitotoxic effects of these. This is a bit more of cell biology, which strengthens the case of this from others, wherein by retinoic acid in a lab in Johns Hopkins, retinoic acid injection caused seizures due to regulator of EAT3. This is a homolog in mice, and that downregulates this particular gene. So there's a link of chemically induced seizures, though not hot water epilepsy, where the link with epilepsy has been shown. The people who have been with us for over 1.5 decades in these studies, Sanjeev Sinha and Prasasti Chandra, Shalini, Rinki, Kalpita and Mohan are students in the lab who have been carrying out this work. I think I'll stop here and take any questions.